The first step in prenatal development is the pre-implantation period. The pre-implantation period takes place during the first week after conception. During conception, the woman's ovum is united with a man's sperm. The result is a zygote. A zygote is a fertilized egg. The final stages of meiosis occur in the ovum at this time, resulting in the joining of the ovum's chromosomes and the sperm's chromosomes. 46 chromosomes are now joined together, 23 coming from the ovum and the other 23 coming from the sperm. The resulting 46 chromosomes is the proper amount, also known as the diploid number, and the 23 chromosomes from each side is known as the haploid number. The zygote now has a reflection of both biological parents. A karyotype is a photographic analysis of a person's chromosomes. The presence of either having XX chromosomes or XY chromosomes makes the sex known. XX chromosomes would result in a female, and XY chromosomes would result in a male. Meiosis only takes place during the fertilization period. After the fertilization stage is complete, mitosis begins. Mitosis is individual cell division. Cleavage is the process of the cell division progressing and splitting into more and more cells. The solid ball of cells is known as a morula, and the zygote is now formed into a blastocyst. The blastocyst continues to split into smaller cells by cleavage during mitosis, making its way to the uterus. Mitosis takes place during tissue growth or regeneration. Once the blastocyst becomes embedded in the endometrium, it undergoes implantation. After a week of cleavage, the blastocyst consists of a layer of peripheral cells, known as the trophoblast layer, and another smaller layer of embryonic cells, known as the embryoblast layer. There are a couple of things to consider during the pre-implantation period. During the process of meiosis, major congenital malformations sometimes occur. Chromosomal abnormality occurs in 1 out of 10 cases, such as Down syndrome. Down syndrome is also known as trisomy 21 because it occurs when an extra chromosome number 21 is present after meiotic division. Widely spaced eyes, mental disability, a flat-bridged nose, and weak tongue muscles are all results of Down syndrome. A child with Down syndrome has a higher risk of periodontal disease, delayed tooth eruption, and fewer teeth present with microdontia. Microdontia is a condition in which one or more teeth appear smaller than normal. Another thing to consider during the pre-implantation period is an ectopic pregnancy. An ectopic pregnancy is a pregnancy that occurs outside of the uterus. Most commonly, it occurs in the fallopian tubes. The disturbance is usually associated with factors that delay or prevent transfer of the zygote to the uterus. Ectopic pregnancies are now able to be treated by medication, but in the past they would often erupt, causing a miscarriage and life-threatening problems for the mother.
We will now enter the embryonic period. The embryonic period begins at the beginning of week 2 and continues until the end of the 8th week. This stage is important because patterning happens, which is a physiological process that is essential in continuing development of an implanted blastocyst to a bilaminar embryonic disc to then an embryo. Within the first week, In the embryonic period, there's many changes. As we can see from Table 3.3, induction, proliferation, differentiation, morphogenesis, and maturation all have very important parts to this cycle of life. Each play an important role for continued development. Induction is the process in which a group of tissue influences another group of tissue to develop. Proliferation contributes by the buildup of cells byproducts, which allows growth. Differentiation is responsible for the change of the identical cells to become different. We also will see the formation of specialized cells which aid in the beginning stages of all major tissue types, organs, and organ systems. There are 3 types of differentiation that affect certain structures within the developing embryo. One being cytodifferentiation, which is the development of different cell types. Another is histodifferentiation, which is responsible for development of different histologic tissue types. And morphodifferentiation, which is accountable for the differing morphology, which makes up the structure and shape of each organ or system. In the second week after entering the embryonic period, the momentum does not slow down. At this stage, the implant blastocyst grows even more by proliferation, which is accumulation of byproducts, subsequently resulting in changes in the cell morphogenesis, which is the development of tissue structure due to migration. Due to the increased cell differentiation, we now have the embryonic cell layer, also known as the germ layer, present. The bilaminar embryonic disc is then developed. As we can see, this constitutes a 3-dimensional, somewhat flattened circular plate of cells. The bilaminar embryonic disc is sandwiched in between the amniotic cavity and the yolk sac, which gives the nutrients to that bilaminar embryonic disc.
In the beginning of the 3rd week, the primitive streak begins to form. This streak is a furrowed and rod-shaped thickening accumulation of cells located in the midline area of the blastocyst. The cell now has bilaminar symmetry. After the formation of the primitive streak, a new layer of embryonic connective tissues forms. The cells from the epiblast layer migrate to the area located between the epiblast and the hypoblast. This migration occurs around the primitive streak, and the newly formed layer is referred to as the mesoderm. The bilaminar embryonic disc now has 3 layers and is now the trilaminar embryonic disc. The trilaminar embryonic disc consists of the ectoderm, the mesoderm, and the endoderm. There is also a cephalic and caudal end. The cephalic end is where the oropharyngeal membrane forms. This is where the digestive tract begins. The caudal end is where the cloacal membrane forms. This is the terminal end of the digestive tract. Here we have a picture of the transition of the bilaminar disc to the trilaminar disc. In the top picture, we can see the primitive streak and the location of the epiblast and the hypoblast. In the bottom picture, the mesoderm has formed. This is the red layer in between the ectoderm and endoderm. In the later part of the 3rd week, the central nervous system begins to develop. A specialized group of cells from the ectoderm, which are called neuroectoderm, localize to the neural plate of the embryo. The neural groove then begins to form as the neural plate deepens and invaginates. By the end of the 3rd week, neural folds surround the neural groove. Neural crest cells from the neuroectoderm disperses from the crest of the folds. These cells are essential for development of the face and neck areas, including most of the tissues in the oral cavity. The mesoderm additionally differentiates and divides into 38 paired cuboidal segments of mesoderm. These segments are located on each side of the neural tube and are referred to as somites. These somites will appear as distinct elevations on the surface of the sides of the embryo. The somites will give rise to the skeletal structures in areas such as the head, neck, and trunk. In the 4th week, the axis is established as the forming tissues are placed into their proper positions. There is now a somewhat tubular embryo. A long hollow tube lined by the endoderm forms from the cephalic end to the caudal end. This will be the location of the digestive tract. The digestive tract will consist of the foregut, the midgut, and the hindgut. With this formation of the digestive tract, 4 pairs of pharyngeal pouches also form from the invaginations on the lateral walls lining the pharynx. By the end of the 4th week, we can see the development of the primitive eyes, the ears, the nose, oral cavity, and the jaw areas.
After the embryonic period, we transition into the final stage of prenatal development, the fetal period. From the completion of 8 weeks of pregnancy until the end of pregnancy, the developing human enlarges to become a fetus or unborn offspring of a mammal. During this stage, developmental changes are occurring to help aid the fetus in independent living outside of the mother's womb. The changes in the fetal period may not be as profound as the changes in the previous periods of prenatal development, but the changes that do occur are crucial in that they allow the newly formed tissue types and organs to function properly, and the embryo changes to a recognizable human being. Some of the important changes that occur consist of the continuation of controlled cellular growth and accumulation of byproducts, changes in the identical embryonic cells to become distinct structurally and functionally, and the development of specific tissue structure or differing form due to embryonic cell migration or proliferation and inductive interactions as previously discussed, along with the continuation of proliferation, differentiation, and morphogenesis. By the end of the 4th month, the unborn child is moving, its heart is beating, and it has grown considerably. At the beginning of week 9, the head is half the normal fetal size, but by the end of week 12, the body more than doubles in size, and the upper limbs reach relatively normal lengths, leaving the lower limbs to remain short and small. One will see a broad face, eyes separated, low ears, and eyelids fused shut. At the end of week 9, the male and female genitalia begin to differentiate themselves, and intestinal loops start moving to the abdomen from the umbilical cord. By the end of week 12, the fetus will also respond to stimuli. Weeks 13 through 16 are periods of rapid fetal growth, where the legs become longer and skeletal ossification progresses rapidly. Early brain development can be seen on the scalp hair pattern, but the head is somewhat small compared to a 12-week fetus. Between weeks 17 and 20, growth slows and lower limbs reach their final relative proportions. During this period, fetal movements are prominent and can be felt by the mother. The eyebrows and head hair are visible along with fine, downy hair that covers the entire body of the fetus. This is a picture of a tetracycline stain on the permanent maxillary anterior teeth as a result of a systemic tetracycline antibiotic therapy of a child during the time of development of the permanent dentition. The antibiotic becomes chemically bound to the dentin, resulting in a yellow to yellow-brown discoloration that can occur in slight, moderate, or even severe cases. Permanent teeth may be affected if the drug is given to a developing child, and the treatment may require a full coverage crown or crowns to improve the appearance of the teeth as shown in the picture. In some cases, teeth whitening can even out the discoloration. A child's deciduous teeth can also be affected if the antibiotics are ingested during the fetal period.